Elise. Elise heads the Gynecologic Cancer Therapeutics section at CTEP. She trained at University of Michigan and received her medical oncology training at the NCI. She spent 2 decades overseeing a laboratory, and that's where I know Elise, in clinical programs at the NCI. And there used to be this thing called angiogenesis, which kind of went out of favor with all this IO thing. We still believe in angiogenesis, but we're not going to have a lecture on that. Elise is now head of the Gynecologic Cancer Therapeutics section at the NCI Cancer Therapy Evaluation Program. She coordinates the development and execution of advanced phase 2 and phase 3 gynecologic cancer clinical trials, and also is involved in overseeing studies in gastroesophageal-pancreatic neuroendocrine tumors. So that's a unique niche and something that we talked about earlier today. Elise has received the Ovarian Cancer National Alliance's Rosalind Franklin Excellence in Ovarian Cancer Research Award, the NIH Director's Merit Awards, and Diversity and Mentoring Awards. Elise is responsible for oversight of the NCTN Core Correlative Sciences Committee, and we are here to learn a bit more about the first few years of this process. Thanks for joining us.
It wasn't 75 years, it was 7.5 years, right, David? Right, okay. So the whole point here, as a unreformed scientist who pushes paper, is the tissue. The tissue really is the issue, and I have to give Lee credit because that was his title. But with that in mind, wanted to just help you see some of the tools that you have where you can move ahead and test hypotheses, because you as an organization have been collecting tissue forever, and there's a lot of it to be used. And one of the things that we recognize is that these unspecified future research opportunities, by saving those tissue in trials, we can move science forward. So we have 2 mechanisms. One is to get biospecimens from phase 1 and 2 trials. Most of those are used up. But if you know that there are some remaining and you want access to them, you just have to send a proposal into CTEP through the standard approach through the PIO, the Protocol Information Office. More importantly, there's access to biospecimens remaining from phase 2, 3, and phase 3 studies that are now in an annotated biospecimen collection resource tool called Navigator. Anything that collected tissue and for which the tissue is available going back to 1996 are all being put into Navigator. Not all the studies are in yet. But it is an inventory resource that you can use, and there's complete clinical annotation because these are from the phase 2, 3, and 3 studies. And so the database that goes to that primary study becomes available for the tissue. So if you just type in NCI Navigator into your browser, you will come up with the web link that's at the bottom of the page. It is a resource for everybody. When we first began the Correlative Science Committee, all the proposals had to come in through the network groups. Now there's a resource that everybody can go in. It does not have to come through the group. We certainly invite you to collaborate with SWOG or your other network group if you're using a study, but it's not a mandate. This is also not a mandate for US-only investigators, although, shoot, you put the work in, you should get to get the samples. And it starts out with you just going to this website. That's what it looks like, and you can go in there and you can search for specimens, and you can see what's available by disease, what's available by protocol. You can see whether your scientific questions could be met by some of those resources. That's all free and easy. If you want to go to the point of submitting a letter of intent for feasibility, you do have to get a CTEP IAMS number. Probably everybody in the room's got one of those already because you're part of SWOG, and in order to be part of SWOG, you probably already have that. And so that's how you get into the next step. And an LOI is not a letter of intent for scientific evaluation, but for feasibility. Ultimately, if it's determined to be feasible, there's a proposal that's submitted and reviewed by the committee. I'll go through this in a little more. And then the expected post-approval elements, if there's intellectual property from a CRADA partner, demonstration that you have IRB and MOU approvals and that you have funding to do what you want to do. So you don't have to wait until you have the funding to come in and ask to do the work, but you can't get the samples until you demonstrate that you have the money. And with everything from NCI, you just have to agree that you're going to make the information, you're going to publish it, and you're going to put the data into the database. As you go further, you can see again, here's for exploring, here's where you put your IM number. This is the flow. As you go through it, you can look up any sort of information. If you know the trial ID or you just want to say, I want to look at melanoma of any sort, type that in, but you can narrow it down. You can narrow it down to the point where there may not be any specimens, but all the parameters are available through that. And you can also find out from trial publications links through clinicaltrials.gov. And as you go through step page by page, you'll find that it takes a little getting used to because there's so much in Navigator to go through. But once you get the feel of it, you can see how you'll be able to say, well, I have this question in this subset of melanoma, or all melanoma, or only MSI this. So those eventually will be the way to do it. So again, the LOI is about feasibility. It's not reviewed on scientific merit. Are the samples there? Is there consent for the use? These are elements that have now, because of your ops program here at SWOG and at the other network groups, that information is findable. So you go in, there's the front door service, so there's a human being if you need that person, where you can communicate with them. Then your request goes to the source of the original trial. So it would come to SWOG Ops and Stats to see what information is there. Then there's a short confirmation that gets sent back to you. So you can see LOI in progress, submitted, feasibility and review, feasibility complete, etc. And ultimately, then, it comes to the Core Correlative Science Committee. I don't know, I think my chief was into C's that day, so it's CCSC. And that is the group represented by many of you in the room, and from all of the network groups, who then come together to review the science and prioritization of use of those irreplaceable biospecimens. They are retrievable. It's been so busy, we actually had to start up with Navigator a second correlative science committee. So CCSCA, co-chaired by Dr. Ellis and also Dr. Michael Beerer, reviews adult solid tumor proposals, and B, which is chaired by Stephen Skapek from COG and John Bird, reviews heme, sarcoma, and pediatric proposals. Pediatrics is coming online shortly, COG. And also does overflow so we can try and not make the delay too great. This on the bottom is the web link, but if you type in NCTN Core Correlative Science, you'll find us also. If you want a sample that's not being used up, for example, you want to use a scan of an H&E slide. If it's already in the system and scanned, you don't even have to go through all of this. If it's not yet been scanned, then it becomes a resource issue and it does have to be reviewed. But there are if there are already other scans involved, there are some areas where you can avoid coming through the committee. The committee has representatives across all of the network groups, also translational scientists, pathologists, clinician scientists, the banks, and a special thank you to Dr. Ramirez, who was our first bank representative, and now JR, who's helping us. Also, statisticians from your group as well as NCI. If the trial was positive, we're going to be persnickety. We don't want to use those samples for anybody's favorite in the back pocket idea, because it's a positive study, and those really, the value of those samples, you can imagine, has a different level. And so for those, it's really ideally used for validation, preferably for prediction, not prognosis. And remember, you cannot predict a prognosis, which we see all too often, but it's got to predict a treatment outcome interaction. Those are the top priority for those samples. And if the drug is dead, the study is negative, a couple other criteria which you could find on the website, then there's more flexibility for exploration and hypothesis-generating questions. But really, this is about ultimately trying to do validation with locked-in assays and parameters. The proposals come in via Navigator. We get them reviewed. We actually send back written questions before the committee meets to give you a chance to answer things that might help the proposal succeed. It's a closed call, and then there are 3 options: approval, revisions, and disapproval. Revisions oftentimes are areas to help not only improve it, but clarify, especially statistical questions. So in 4 years, we've now had almost 120 submissions. You can see the breakdown by the group, and then at the bottom in Navigator, since Navigator's been open April 2018, and those are not group-specific submissions, although many have group collaboration involved. And you can see the breakdown also here of the 74 that went on to be reviewed. You can see the breakdown by disease site. And of those 109 at the time we did the analysis, 39 had been approved, 25 have been disapproved, a few withdrawn after revisions. You wonder, what is not forwarded for review, and why did almost a third? That meant that when the proposal came in, it was missing key elements. Maybe there was no assay description. The statistics weren't missing in action, or didn't fit the hypothesis. So at NCI, we review to make sure that everything is in the proposal so that our committee has the ability to really assess the science. If it's not there, we'll send it back and say, hey, please do these things. That's not a nerf. That's a just tidy it up and send it back in. It's neither a yes or no. It's just not enough information. Not everybody sends it back in, which is unfortunate. Again, here you can see 74 have been reviewed. The bulk of them have actually been approved ultimately. Most of the ones that come back for revisions come back and are approved. And that has been increasing over time. Of the approved, we've had 62% remain active right now. So in the 4 years since the committee started, only 15% are complete. One of the reasons is, of course, if you're getting an approval but you haven't got funding yet, it takes time to get the funding, then it takes time to get the samples. So we realize that there is a built-in set of delays between the initiation of the idea and the execution of the laboratory work. To date, we have documented 3 publications, 4 abstracts, with 5 more submitted. But many of these come in with a clinical trial number and not the core correlative science number. And when you try and query a database and you don't know what to query it on. So I think that our success is a little bit higher, and we just sent out another round of 3-year progress review questions to see how many more of the approved studies of that 62% are active and in completion. I think the most important thing is, and at least to me, if I were sitting where you is, it's how can I get in there? How can I get those samples? And how can I be successful the first time around? So I showed you how to find out what's there using Navigator, how to parse out what samples might fit the questions that you want to ask, how you go about getting a feasibility review. It's possible that you want to do something that can today still only be done in immediately processed, fresh, frozen material, and it doesn't exist. Well, that's disappointing, but that just means write a study where you make sure you can get those things done. So you are limited by what we have. And obviously, if it's something that has a freeze-thaw problem, that also is to be taken into consideration. So there's a limitation by what's available and the numbers, but it's really a tremendous resource. Once you get feasibility that says that to move ahead, then the question is, how can you do that successfully? And so one of the things that frustrated me as an investigator, and I'm sure many of you, especially if you're a young investigator or you've mentored someone, is that it always helps to know where the pitfalls are, right? Not just when you write them in the grant, but how to prevent them from happening when you're writing the grant or proposal or clinical trial. So I went back to look at what were some of the things that caused a no initial review, or disapprovals, or frustration on the part of my committee. And I can tell you, this committee is pretty amazing group of colleagues, and they work very hard to find the silver lining in some of these pretty dark clouds, JR's laughing, because it's true. Probably the biggest frustration, and I remember being on the ASCO grant committee, and it was the same thing. There was no statistical plan. If you actually look at our proposal form, it has all of these prompts in it. It really lists, 8 or 10 different things that it wants in the statistical section and in the assay section. So I do encourage you not to skip over that, because we're trying to give you what you need for success. But the biggest fallout is a statistical section that's incomplete, where the plan cannot be recapitulated by the reviewing statisticians, or there's no way that there's power to get anything out of it. 50 samples. I only want 50 samples, but I'm going to do a prediction analysis of treatment by outcome, which is usually in the hundreds. Well, I'm only targeting a differential of 6.0. Well, nothing's going to have, most of the things you read, you see an odds ratio of 1 point something or 2 point something. So if it comes in, you're only using 50 samples because you're targeting an odds ratio of 6, you should realize there's probably, not always, a problem there. So that's probably one of the biggest frustrations that the committee runs into. And then assay details, especially if you're going after samples from a positive trial where you need to be validating, you need to have a locked down discriminant. Because validation is, you're asking, can we make an assessment on a case-by-case basis? Which means you can't have above the median and below the median, because there's no median, right? It's a case. So locked down parameters that can be applied in a case-by-case basis if you're going after a validation. We want to know what assay you're using. Just because the company can do it with a quality control of 98% doesn't mean you can. And anybody who's been in the laboratory, and I will admit definitely from all my years in the lab, I can't always repeat my own things, right? Because some days the antibody's dead and you don't realize it. But let's be honest, it's important to know the assay details and how they work in the institution doing the analysis for the project. That's what we mean when we ask you that. We want to know what are the QC parameters, the quality parameters in your hands. And occasionally we see objectives that don't fit the hypothesis or the design, or someone really wants to do a lot of exploration, so they put in a primary objective they think will fly by so that they can get access to the samples and do other things. And the folks that we have on our committees are really, really good, and they pick up on that. So these are the things. Don't hesitate to reach out. You have an outstanding resource here with Lee, Dave, JR, others in the room who are part of the program. You're always welcome to reach out to me as well before you send something in. We're happy to help out with questions. We're happy to be a resource if you're not sure how to go about it. I think Navigator, it looks daunting at first, but it actually, I could get through it, and if I could get through it, trust me, you can. And also, I have here our general email address and Courtney McCoppin, who is our administrator for the group. If you go to our website, on the bottom there is an updated quarterly, the list of what's been approved, because that can be in the public domain. So you can also see if there's something that's already pending that may be related to your hypothesis. It has the investigator. It's always an opportunity for collaboration as well. Ideas that are not approved don't get listed. If you're unsure if a study is available if it's not Navigator and you don't know why, reach out to us. That's probably the most common question we get. It may be that the study hasn't been completed and reported yet. So this is only for those samples that are left over after. This is that resource. That's why I told you went back to 1996. So if a study is ongoing now, that's not a resource. It's not going to come through this mechanism. It's not available to the general public. However, if you have a great idea, speak with the study team and the study translational scientists as to whether or not that idea could or should be put in by amendment as part of the actual clinical trial. These are all for post-trial questions, and they are independent of what has been approved as an integrated endpoint to the clinical trial. So you don't need to come in through this mechanism to request samples when you've already got approval for an integrated endpoint. This is to amplify your science, not to restrict it. Thank you.